Thank you, Ron, for the introduction, and thank you, the organizers, for the invitation. Marker Therapeutics is a public trading company, so this is just a disclaimer that the lawyers ask me to put every time, so please, more information, please visit the website. We have developed quite an interesting technology. Some of you might be familiarized with CAR T cells, and what we do is actually something similar that address many of the challenges currently faced with adoptive T cell therapy and CAR T cells. If you look in the lower left-hand side, you basically see that genetic engineering definitely has validated the space for adoptive T cell transfer. However, it's now well recognized that the single target antigen can result in the tumor evolution and adaptation that leads to the resistance. What we actually have developed is immunological transfer of T cells that are educated in the laboratory without genetic engineering. Our final drug product constitutes of thousands of individual alpha-beta TCRs that is capable of recognizing hundreds of different epitopes. We have tested this technology in over 200 patients. We have seen a very favorable safety profile. And we have seen early data of efficacy in both hematologic malignancies and solid tumor. This technology was developed at Baylor College of Medicine, and the company, we have the exclusive worldwide license for this approach. Under the company umbrella, and I would like to acknowledge that working with FreeMind, we have been quite successful, and we have anchored over 17 million in non-diluted funds from CPRED, FDA, and NIH. And even though we're a publicly trading company, I think that having funding from the government and the agencies is not only a great avenue to advance the program, it's also a huge validation for the technology. And you also get important feedback from scientific and clinical peers. Under the company umbrella, we're advancing 3 different clinical programs. We have a product called MT401 and MT601. We have a program in lymphoma, AML, and pancreatic cancer. One of the programs that I'm the most excited is about our lymphoma program, which we launched recently. This is a phase 1 clinical study. And the reason for that is because I believe that this program has the potential to yield approval in a short period of time, and I will explain why. If you're familiar with the space, you probably also recognize that lymphoma is extremely crowded, and multiple companies are developing therapies in lymphoma. What we're doing is slightly different, and we're basically targeting this product in patients that are CAR failures or CAR relapse patients. And in that way, having the opportunity to develop the product in a clinical population that has a high unmet medical need. Now I'm going to show you 3 pieces of independent evidence that suggest why this clinical study should yield a positive clinical outcome. The first one, it comes from a pre-existing clinical experience at Baylor College of Medicine where we developed the product. And here you can see multiple things. The first thing is that we have evidence that this therapy works not only in Hodgkin lymphoma, but also in non-Hodgkin and non-Hodgkin lymphoma. And here you basically see that this therapy can be given to patients without lymphodepletion. We haven't seen any cytokine release syndrome nor neurotoxicity, and this therapy can convert patients to a complete response. And in the top right-hand side, you see a CT scan of one of these patients responding to the therapy. We also have modeled in the laboratory the response that our therapy has to these type of tumors. Now, let me walk you through this data set. What you see here is this is in the y-axis, this is number of tumor cells, and in the x-axis, time. Now, as expected in the absence of any treatment, you see here I don't know that I have a pointer. Okay, in the absence of any treatment, you basically see the black line. As expected, the number of tumor cells grow very quickly. If you administer CAR-CD19, as expected, you now see a control in the tumor growth. However, if you monitor those cells long enough, you basically see that tumor cells start to appear from that initial treatment. And if you examine further those tumor cells that appear, you basically identify that those clones of tumor cells that are becoming resistant, in fact, they lack the expression of the target antigen CD19. And that's something that it makes sense, right? So you have a CAR CD19, it eliminates the tumors that are CD19 positive, and as a consequence, you have the emergence of a different tumor cells lacking the target antigen. And if now you take these residual tumor cells and treat them again with CAR CD19, you basically see, as expected, that these tumor cells now are resistant to future treatments. In contrast, if you take these cells and you treat them with our product, MT601, you can basically see that our cells and our therapy can actually eliminate tumor cells that are even CAR refractory. Now, most recently, we actually have launched this study into the clinic, and here we now are reporting the results from our first patient enrolled and treated in this study. This is a 57-year-old female patient diagnosed with DLBCL. This patient has 4 prior lines of therapies, including CAR CD19, and this patient relapsed to CAR CD19 after 90 days of therapies. After this, the patient was enrolled in the clinical study, where received 2 doses of our therapy. After this, we observed that the therapy was well tolerated. There was no cytokine release syndrome, nor neurotoxicity. 8 weeks later, the patient had a complete metabolic response, and now the patient has surpassed 6 months and is still in complete response. So even though this is very early clinical data and we continue to collect, it basically supports our thesis that MT601 could provide a clinical benefit in patients that are CAR relapse. We will continue collecting data. We have a currently ongoing clinical study called Apollo, and we anticipate to read additional clinical data later this year. Now, I'm going to present what we basically consider will be the future of the company and how we can basically take this technology beyond. And I'm going to finish with presenting just a glimpse of 2 additional clinical programs that we are advancing. This is an off-the-shelf program using a product that we call MT401 in AML. One of the things that probably you can recognize is that a significant bottleneck in the industry is the individualized production of these type of products, right? And even though we have a simple manufacturing process, one way to overcome this is the generation of a pre-existing cellular inventory that can be given to patients as they actually need it. In order to do that, our solution is quite simple. We identify donors, healthy individuals of a particular HLA typing that allow us to pre-manufacture this product. And then when the patient, we manufacture the product exactly the same way. And once these products are then cryopreserved, and when the patient requires the product, we simply perform a partially HLA matching that allows now to pair our product inventory with the patient. So it's a very simple solution to a problem in the industry related to the individualized production of T-cell products. So we're very excited about this program and anticipate to dose our first patient this year. Now I'm going to finish with another program that is very exciting, and is the MT601 program in pancreatic cancer. One of the issues that we have seen in the field is the limited clinical efficacy of CAR T cells, particularly in the context of solid tumors. But this technology is actually very different. We actually have already clinical proof of concept data in pancreatic cancer. This is a phase 1 clinical study that we conducted at Baylor. And here we see this is a combination chemotherapy regimen as a first-line therapy. We basically see here these patients were able to obtain 1 complete response and 3 partial responses after the administration of the cells. Now, this is a very early clinical data, but we will continue advancing this program under the company umbrella and hope to identify whether the administration of our product can in fact have an improved overall survival in patients with pancreatic cancer. So some of the corporate highlights is we currently have a runway that takes us all the way to the end of 2025. We have more than 17 million in cash. We have been awarded more than 17 million in non-dilutive funding. It has been really a great collaborative experience with FreeMind. And we have, as I mentioned before, demonstrated that our therapy is well tolerated, and we have some early signs of clinical efficacy in both hematologic malignancies and solid tumors. And we will continue working this year to collect additional data that hopefully will pave the way to a future approval in lymphoma. With that, I would like to thank the organizers for the invitation, and very importantly, the grant agencies, NIH, FDA, CPRED, which is a great source of funding for companies in Texas, and all our patients and the clinical collaborators and the Marker team. And if there's any questions, thank you very much. Sure.
Your multi-TA, was that like an AND gate or was it OR gate?
Okay, so I'm going to repeat the question. So the question is if this technology is an AND or OR gate. It's a great question, and it allowed me to clarify that we do not use genetic engineering, right? So it's a very simple process. We basically use over a set of peptides, hundreds of peptides that basically amplify, multiple different epitopes from 6 selected tumor-associated targets, right? So the manufacturing process, basically, we take the apheresis material from the patient. We use this peptide library, which is the same for every single patient. We use that peptide library with cytokines that allows the amplification of tumor-reactive cells. As a consequence, you have thousands of individual alpha-beta TCRs that get amplified, targeting hundreds of different epitopes, right? So it's a very simple technology, but it's old school. I guess the element that it is new is the fact that you're basically targeting multiple different epitopes throughout the same manufacturing process. So if you were then to do the same manufacturing process from multiple different individuals, you basically will see a different makeup on the T-cell clones and the target antigens due to the diversity of HLA typing and the antigens expressed by the tumor. But that diversity within the product itself is what makes this therapy unique, or making up an appropriate match for the patient's own tumor characteristics. Perfect. Thank you. Well, thank you for the attention. Thank you very much.